Hi guys, so today we're going to look at some of the key terms that are applicable to what we were, to our little penny flipping experiment on Friday. So how did Mendel study heredity? You guys probably have heard this a couple times before, but what I want to establish is some key words. So heredity is the transmission of traits from one generation to another, and genetics is simply that, the study of, the scientific study of heredity, and that's what Mendel was doing. He actually was looking at pea plants in order to find ways to improve the harvest for the monks in the Augustinian monastery where he was doing his research. Okay? And so what we're going to do today is do some plants and a plant experiment where you have certain pea generation plants. Pea generation just means the parent individuals from which offspring are derived in the study of inheritance. So for Mendel, those parents were purple flowered and white flowered plants, and today you're going to see our fast glowing brassica, and we're going to use certain ones for our experiment. Mendel used true breeding plants. So true breeding means that organisms for which sexual reproduction produces offspring with inherited traits that are identical to their parents. So if this purple self-fertilizes, it still undergoes sexual reproduction. It just so happens that it's doing it with itself. It produces many different sperm gametes and it has many different ovum or eggs to take place. You still have the randomization of fertilization, but for all intents and purposes, the traits will be identical. Okay. So these are important to use to enable to control the genetic product in your hybrid experiment. A hybrid is the offspring of two individuals of different species or two distinct true breeding parents. These parents must have one or more different traits, and the resulting individual will be heterozygous for one or more genes. Okay? So to do the hybrid, you have the purple and the white flower, and the F1 generation will be the hybrids, the offspring of the parents that have one or more different traits. And so what I want you to do now is pause the video if you want to finish writing down your definitions. Okay, you've unpaused. Let's continue. So what does that mean, the F1 generation? So we've got our parental generation, okay? And through a cross, Mendel was able to mate two sexually reproducing individuals. It's just a process that controls mating of individuals in genetic experiments. To really do a good genetic experiment, you need to control your crosses. You need to know who your parents are so that you can interpret the frequencies or the outcomes of the experiment. In the F1 generation, which would be the offspring of the two parents, and the F just stands for philia, and I think that is Latin for children, right? Son or daughter, philia and philias. And then the F2 generation is going to be if you mate two F1 plants together or two F1 individuals, you can get the F2 generation, and you can get F3, and so on and so forth. Mendel made lots of crosses. Not only did he do flowers, but he did flower position, whether it's up high or down low. He did seed color, shape, pod, and through all of these different crosses that he did, and counting the frequencies of the offspring phenotype in the F1 and the F2 generation, he was able to infer certain rules of Mendelian genetic inheritance. So if you need an extra second to write down these terms, please do. Okay, and so once we do our crosses, right, Mendel came up with this law called the law of segregation. What does that mean? Well, here's what he did. He had true breeding purple flowers, true breeding white flowers, and he crossed them. All of the gametes for the purple flower will be will all have the dominant P. All the gametes for the white flower will have the recessive P. When you mate them together, you get your hybrid, which is heterozygous for both. Now this heterozygous, when it self-pollinates itself, or when you pollinate it with another purple plant, this is called a monohybrid cross. It's a monohybrid cross because it's an experimental mating of individuals that are heterozygous for the character being followed, or the self-pollination of a heterozygous plant. Right? So you can self-pollinate that plant or mate that purple plant with another with another hybrid that's heterozygous for the same traits. Mono meaning one, hybrid meaning the offspring of two parents that had different traits that you're interested in. Okay. Now, when we look at the segregation of the alleles during meiosis, half of the gametes will have the large p, and the other half will have the small p, or the recessive trait. How do we know that to be true? Well, if you assume that they segregate independently, okay, and you do your Punnett square, you put the gametes of the male on the top, or the male on the top. So the F1 plant's gametes, you're going to have half being dominant, half being recessive. And on the y-axis, you have from the egg, if it's also similarly heterozygous for flower color, you will have half the gametes being dominant, half being recessive. And when you do your Punnett square, you can see that 3 of them will look like the dominant trait, the dominant phenotype. Of course, 2 of them are heterozygous, but you can't tell that by just looking at the plant. And a quarter of them will be this. If they didn't segregate independently, the frequency of this would be much different. So what Mendel did was, because he saw this repeatedly in all of his crosses, he came up with a law of segregation. And it's a general rule in inheritance that individuals have 2 alleles for each gene. And that when gametes form by meiosis, the 2 alleles will separate, each resulting gamete ending up with 1 allele for each gene. So if we look at this in meiosis and we have our 2 homologous chromosomes that contain the dominant and the recessive trait, you go through interphase, you replicate them, okay, so the recessive is replicated, and then when you go through meiosis 1 and 2, each of these would be pulled apart, and half of them will have the dominant and half of them will have the recessive. So there's a little pictorial version of what's happening there. Okay. And I think it would be very useful that you copy this down, and as you do, think about what this means in terms of meiosis and how the segregation of these alleles into different gametes is what gives you this genetic recombination. Because of course this will happen also in the female, and you can count the offspring, and that's what we're going to be doing later on this week after we plant our seeds. We're going to try to figure out what the frequency is of crossing the F1s together to get our F2 generation. It also says something specific about what our F1s will look like. We'll come back to that later. Well, Mendel wasn't just interested in one phenotype. He was interested in looking at multiple phenotypes. Okay, now this is a lot to kind of take in, but let's look at this. So what would happen if he crossed plants that differed in both seed shape and seed color? You guys did this last week on Friday. Okay, so you had a true breeding yellow smooth, a true breeding green rough, and this is recessive, this is dominant. Okay. And when you cross those and you get your hybrid, your F1 generation, you're going to notice that you have it being heterozygous for both the traits. Now it's going to look, even though it's heterozygous, it's going to look just like the true breeding dominant, because of course it's going to have both dominant alleles. Now, Mendel noticed right off the bat when he did his next cross, which he called a dihybrid cross, which is an experimental mating between individuals heterozygous for both of the 2 characters. Right? So it's heterozygous for the color and heterozygous for the texture of the seed for both the 2 characters or the corresponding self-fertilization. So he allowed self-fertilization of this plant. And he said, hey, if I look at the frequency of the offspring, I can probably figure out if both of these genes are on the same chromosome or not. Well, he didn't know about chromosomes, but he had a sense of the true nature of how they combined. So there's 2 predictions. One is that if the 2 genes are on the same chromosome, 25% of them should be rough, because each of the gametes, there's only 2 ways to split these gametes, right? You're going to have R and R and Y, or R and little y. I'm sorry, you're going to take this gamete and split it to here. You can only have, if these are on the same chromosome, then when you go through meiosis, you're just going to have those chromosomes. So, if it's inheriting the RY on the same chromosome, then when it goes through meiosis, the RY will still be on the same chromosome, assuming that there's no genetic recombination, which we'll get into later on in the week. Or what if they do? What if the offspring inherit each of these alleles, or each of these genes, because they're on different chromosomes? So, for instance, if R is on one chromosome and if R is on one chromosome and Y is on the other, what would that give? Well, you have to look at the different combinations, right? So if the alleles sort independently, then you can get a capital R and a uppercase Y. You can get a capital R and a lowercase y. You can get a uppercase Y and a lowercase r. You can get a lowercase y and a lowercase r. And notice that there's an assortment of gametes. Depending upon how you combine the different chromosomes as they separate through meiosis, you get an assortment of gametes. And so what Mendel called this was the law of independent assortment. This is a general rule of inheritance that when gametes form through meiosis, each pair of alleles for a character segregates independently of other pairs. He knows this to be true because these predicted gametes, when crossed with a similar hybrid in this dihybrid cross, you get characteristic frequencies of each of the phenotypes. 1/16 green and wrinkled, 9/16 rounded and yellow. I think it would be beneficial afterwards, after watching this, to look at your results from this. Did your results give the similar amount? And we'll go over that in class right after the warm-up. And finally, how do we deal with the fact that you have these heterozygotes? How did Mendel know that these were these hybrids were heterozygous? Well, you can do what's known as a test cross. For instance, this black fur color is dominant. Is this black homozygous for the black trait, or is it heterozygous? Well, to do that, you can do a cross with a homozygous recessive brown dog. Right? So if for instance, this was dominant, then this genotype would only give you one type of gamete that has a B, and of course this will only have one type of gamete, which is the recessive trait. But if it's heterozygous, notice that you can have two different gametes. And so if the offspring of this test cross is 50% black, 50% chocolate, then you know that this is heterozygous and not homozygous. So a test cross is the mating between an individual with an unknown genotype and an individual that is homozygous recessive for that same character. Okay, so these are some terms. There are a lot of terms. I want you to know them. We did some practice on Friday. We'll continue to do that today in the lab.